Hello, and we're moving on to 11.4 meiosis. I know you've been waiting a long time for meiosis, and here it is for you. So our goal is to describe the process of meiosis, including independent assortment and crossing over, and explain how reduction division results in the formation of haploid gametes or spores. And maybe you've seen this one already from this far side today is although it lasted only 2 million years, the awkward age was considered a hazardous time for most species. Alrighty, let's move along here. I have on here a quick look at some of the terms that we've learned already. So the principle, these are the Mendel's principles. Principle of segregation. 2 factors, alleles, for a trait separate during gamete formation. Principle of independent assortment. Factors of a trait separate independently of one another during gamete formation. Another way to look at this is whether a flower is purple has nothing to do with the length of the plant stems. Each trait is independently inherited, genes for different traits. The principle of dominance and recessiveness is one trait is masked or covered up by another one. Alrighty. So our objectives is to contrast the number of chromosomes in body, cells, and gametes, summarize the events of meiosis, and contrast meiosis and mitosis, and describe how alleles of different genes can be inherited together. So let's think about it first. As geneticists in the early 1900s applied Mendel's laws, they wondered where genes might be located. They expected genes to be carried on structures inside the cell. But which structure? What cellular processes could account for segregation and independent assortment, as Mendel had described? So objective one is contrast the number of chromosomes in body cells and in gametes. So what I mean by that is how many cells or chromosomes we have in our regular cells all over us, and then our specialized gamete cells, just for reproduction purposes, those guys. So chromosomes, as you know, those are the strands of DNA and protein inside the cell nucleus. They are the carriers of genes. The genes are located on specific positions on the chromosome. So this is an example of a chromosome before it's been duplicated, and these genes are all along the whole length of the chromosome. So the body cell in a fruit fly has about 8 chromosomes, as you can see from this figure right here. 2, 4, 6, and these 2 little ones are 8 here also. 4 of the chromosomes come from its male parent, and 4 come from its female parent. So 1 from the male, here, here, and here, and matching 1 from the female, here, here, and here. All right? These 2 sets of chromosomes are homologous, meaning that each of the 4 chromosomes from the male parent has a corresponding chromosome from the female parent. A cell that contains both sets of homologous chromosomes is diploid, meaning it has 2 sets, 1 from the daddy, 1 from the mommy. The diploid number of chromosomes is sometimes represented by the symbol 2n. 2n because it's double the number, right? For the fruit fly, the diploid number is 8, which can be written as 2n equals 8. So then n represents twice the number of chromosomes in a sperm or egg cell. So n would be obviously be 4. 1, 2, 3, 4 of different types of chromosomes, but they have a matching pair. Some cells contain only a single set of chromosomes, and therefore a single set of genes. Such cells are haploid, meaning 1 set. The gametes of sexually reproducing organisms are haploid. So for fruit fly gametes, the haploid number is 4. You get 1 from your daddy, like this one, and this one. All right? And n could be 4 then. So objective 1 is contrast the number of chromosomes in body cells and gametes. Well, for every organism, it's going to be different. But the diploid cells of most adult organisms contain 2 complete sets of inherited chromosomes and 2 complete sets of genes. Gametes, on the other hand, contain a single set of genes and are called haploid cells. That's going to be important moving forward. So let's summarize the events of meiosis. Here we go. Here's meiosis for us. So meiosis is a process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Meiosis usually involves 2 distinct divisions called meiosis 1 and guess what? Meiosis 2. By the end of meiosis 2, the diploid cell becomes 4, bing, 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 haploid cells. Start with one of these, and it goes through all these series of divisions and end up 4 haploid cells. Got it? Let's start one step at a time then. And I left the writing up here for you to help you follow along easier too. So just prior to meiosis 1, the cell undergoes a round of chromosome replication called interphase 1. So the chromosomes are replicating themselves. Each replicated chromosome consists of 2 identical chromatids, just like we know from mitosis, joined at the center. The homologous chromosomes pair up, forming called a tetrad, which contains 4 chromatids. Let's move here. Okay, the homologous ones pair up and form a tetrad. As homologous chromosomes pair up and form tetrads, they undergo a process called crossing over. First, the chromatids of the homologous chromosomes cross over one another like this. See that? They pair up and they start crossing over. Then the crossing sections of the chromatids are exchanged. You can see how that happens right there. The crossing over is important because it produces new combinations of alleles in the cell. Then we move on to metaphase anaphase. As prophase 1 ends, a spindle forms. Remember the spindles right here, like mitosis, and pull each homologous chromosome pair toward opposite sides of the cell. All right, like in mitosis. And when anaphase 1 is complete, the separated chromosomes cluster at opposite ends of the cells. All right, the 2 new cells. So now, would I move on too much? I think so. So meiosis 1 results in 2 cells called daughter cells. Back up. During telophase 1, a nuclear membrane right here forms around each cluster of the chromosomes. Here's a nuclear membrane. Cytokinesis follows telophase 1, forming 2 new daughter cells. There we go. So meiosis 1 results in 2 cells called daughter cells, okay? Each of which has 4 chromatids, as it would after mitosis. Because each pair of homologous chromosomes was separated, neither daughter cell has the 2 complete sets of chromosomes that it would have in a diploid cell. The 2 cells produced by meiosis have meiosis 1 have sets of chromosomes and alleles that are different from each other and from the diploid cell that entered meiosis 1. So you have different kinds of chromosomes. The 2 cells produced by meiosis 1 now enter a second mitotic division. Unlike the first division, neither cell goes through a round of chromosome replication before entering meiosis 2. So that's important. You're not going to duplicate our chromosomes here. All right? So as the cell enters prophase 2, their chromosomes consisting of 2 chromatids, all right, become visible. The chromosomes do not pair to form tetrads this time, though, because the homologous pairs were already separated during meiosis 1. So during metaphase of meiosis 2, or metaphase 2, chromosomes line up in the center of each of the cells. They line up like metaphase, but in mitosis. And then as the cell enters anaphase, the paired chromatids separate. In the example shown here, each of the 4 daughter cells produced in meiosis 2 receives 2 chromatids, little chromatids, each of them. These 4 daughter cells, 1, 2, 3, 4, now contain the haploid number N, just 2 chromosomes each. So the haploid cells produced by meiosis 2 are called gametes. In male animals, all right, these gametes are called sperm. Sperm cells, the male gametes, are mature spermatids that have already undergone differentiation. So what happens is they start out like the little cell, like you can see right here, and they go for differentiation, and this process is called spermiogenesis, as a matter of fact. And then that's when you get the flagellum, you get reduction of the cytoplasm, an increased number of mitochondria. You need lots of mitochondria to power those little buggers, all right? And in some plants, over here, pollen grains, spores, contain haploid sperm cells. So, and also in female animals, generally only one of the cells produced by meiosis is involved in reproduction. The female gamete that we get from meiosis is called an egg in animals and an egg cell in some plants. And we'll be learning about the plant reproduction cycles later on in other chapters. So when you have the sperm the male gamete and the female gamete, they meet, then you have fertilization, which is the fusion of the male and female gametes. And this generates a new combination of alleles called a zygote. All right, gametes and zygotes. And then the zygote undergoes the cell division, mitosis, and eventually forms a new organism. So let's summarize the events of meiosis. In prophase I of meiosis, each replicated chromosome pairs with its corresponding homologous chromosome. During metaphase I of meiosis, paired homologous chromosomes line up across the center of the cell. And during anaphase I, spindle fibers pull each of those homologous chromosomes paired toward opposite ends of the cell. In telophase I, a nuclear envelope forms around each cluster of the chromosomes. Cytokinesis follows telophase I, forming those two cells. All right? And as the cells enter prophase II, their chromosomes, each consisting of 2 chromatids, become visible, and the final 4 phases of meiosis II are very similar to those of meiosis I. However, the results is 4 haploid daughter cells. So from one cell, you get 4. Yes, not 5, 4. All right, let's compare and contrast meiosis to mitosis then. So, mitosis is a form of asexual reproduction, whereas in meiosis is an early step in reproduction. There are 3 other ways in which these two processes differ. In mitosis, when the 2 sets of genetic materials separate, each daughter cell receives 1 complete set of chromosomes, as you can see. All right. In meiosis, homologous chromosomes line up and then move to separate daughter cells. As a result, the 2 alleles for each gene segregate from each other and end up in different cells. Mitosis does not, the sorting and combination of genes in meiosis result in a greater variety of possible gene combinations than could result from mitosis. Mitosis does not normally change the chromosome number of the original cell. You wouldn't want that to happen because then your cells will be all messed up. You gotta have the same number in all of them. But meiosis does. It reduces the chromosome number in half. On the other hand, a diploid cell that enters mitosis with 8 4 chromosomes will divide to produce 2 diploid daughter cells, each of which also has 8 4 chromosomes. On the other hand, as I was going to say, the diploid cell that enters meiosis with 8 4 chromosomes will pass through 2 meiotic divisions to produce 4 haploid gamete cells, each with only 4 2 chromosomes. All right. Mitosis is a single cell division, resulting in the production of 2 genetically identical diploid daughter cells, as you should know. But meiosis requires 2 rounds of cell division, and in most organisms produce a total of 4 genetically different haploid daughter cells. So when we compare meiosis and mitosis, in mitosis, when the 2 sets of genetic material separate, each daughter cell receives 1 complete set of chromosomes. In meiosis, homologous chromosomes line up and then move to separate daughter cells. Mitosis does not normally change the number of chromosomes, number of the original cell. This is not the case for meiosis, which reduces the chromosome number by half. Mitosis results in the production of 2 genetically identical diploid cells, whereas meiosis produces 4 genetically different haploid cells. Objective 4, moving on, describe how alleles from different genes can be inherited together. Thomas Hunt Morgan's research on fruit flies led him to the principle of gene linkage. After identifying more than 50 Drosophila fruit fly genes, Morgan discovered that many of them appear to be linked together in ways that seem to violate the principle of independent assortment. For example, Morgan used a fly with a reddish-orange eyes, these guys like we have in our classroom, or we had, in miniature wings in a series of test crosses. His results showed that the genes for those 2 traits were almost always inherited together. So you always get orange eyes and miniature wings together. Only rarely did the genes separate from each other. So Morgan and his associates observed so many genes that were inherited together that before long, they could group all the fly's genes into 4 linkage groups. The linkage groups assorted independently, but all the genes in one group were inherited together. As it turns out, Drosophila has 4 linkage groups and 4 pairs of chromosomes. All right, Morgan's findings led to two remarkable conclusions. First, each chromosome is actually a group of linked genes. All right. Second, it is the chromosomes that sort independently, not the individual genes. Alleles of different genes tend to be inherited together when those genes are located on the same chromosome. So by doing all these tests on these Drosophila, he could actually determine where the genes were on the chromosomes. Interesting. So moving on, in 1911, the Columbia University student Alfred Stuyvesant, well, I don't know if I'm saying that right, wondered if the frequency of crossing over between genes during meiosis might be a clue to the gene's locations. Stuyvesant reasoned that the farther apart two genes were on the chromosome, the more likely it would be that a crossover event would occur between them. So like on the ends up here, crossing over would happen most likely. All right? If the two genes are close together, like down here, then crossovers between them would be rare. If two genes are far apart, then crossovers between them should be more common. By this reasoning, he could use the frequency of crossing over between genes to determine their distances from each other. Stuyvesant gathered lab data and presented a gene map showing the relative locations of each known gene on one of the Drosophila chromosomes. All right, here we go. Chromosome 2. All the known genes, purple eye, light eye, vestigial, small winged, curved winged, all those. Stuyvesant's method has been used to conduct gene maps ever since this discovery too. So objective 4, describe how alleles from different genes can be inherited together. Alleles of different genes tend to be inherited together from one generation to the next when those genes are located on the same chromosome. All right, so I know that was a lot, and we will review this in class and practice with it too. So objective 1 is contrast the number of chromosomes in body cells and in gametes, summarize the events of meiosis, contrast meiosis to mitosis, and also describe how alleles from different genes can be inherited together. And that definitely all supports describe the process of meiosis, including independent assortment and crossing over. Explain how reduction division results in the formation of haploid gametes or spores. All right, that was a lot. Let's practice. Where's my pretty picture? I forgot. Sorry.